Kind of from this middle table, I've accumulated a series of models that you guys should know, and I'm going to kind of run you through how to approach studying things like the blood vessels and the lymphatic vessels and lymphatic system. The special senses, I'm sorry there wasn't more on there, but it is what it is. Right? I did not think about you. Our special sense is going to be on the practical.
Yes.
They are. Right? We just went over and we had the eye dissection. We had a lot of stuff in terms of the cardiovascular system that I really wanted to cover. Right? So, everyone with me?
Yes.
Yes. So, identify the gland shown in the image.
Thyroid.
It's definitely the thyroid gland. And how do you know it's the thyroid gland? So let's run through the series of questions I could ask. I could ask, identify the structure indicated by the bracket. That is a follicle. Identify the cell type indicated by the pointer. Follicular cells. Identify the structure or the substance indicated by the pointer.
Colloid.
That would be the colloid. So people sound like they did that really well. And then what hormone is produced here? T3 and T4, which is another way of saying thyroid hormone. One's an active and one's an inactive form, triiodothyronine, tetraiodothyronine. It just T3 and T4 pertain to the number of iodine molecules that's pinned to the hormone itself, and it dictates whether it's active or not. So, you guys did that really well. And what is the function of thyroid hormone in general? It increases metabolic rate, right? It bumps up metabolic rate. It increases metabolic pathways like glycolytic pathways. It increases the rate at which glucose is broken down. It increases a lot of things. So, everyone good? Identify the structure indicated by the bracket. So, in terms of histological images that you need to know for the upcoming practical, what are the two glands, or actually the three glands that you really need to know histologically? There's the thyroid gland,
Pituitary.
the pituitary gland, and you need to be able to distinguish between the
Pars.
adenohypophysis and the neurohypophysis, and the pars distalis, which is the glandular tissue that makes up the adenohypophysis, and the pars nervosa, which is the nervous tissue that makes up the neurohypophysis. Adenohypophysis is a fancy way of saying anterior pituitary gland. Neurohypophysis is a fancy way of saying posterior pituitary gland. What's the other gland that you need to know histologically? The so we do definitely need to know the pancreas. And what are the two things about the pancreas? The islets of Langerhans. And there are two cell types embedded within the islets of Langerhans. What are they? Alpha and beta pancreatic cells. Beta pancreatic cells produce insulin, and alpha pancreatic cells produce glucagon. Insulin does what to blood glucose levels? Lowers blood glucose levels. It tells cells essentially to suck glucose up. And then glucagon does what to blood glucose levels? Raises. It tells the liver to start breaking down glycogen, which is the storage form of glucose. Everyone good? What's the other one? The adrenal gland. And specifically being able to tell me the layers of the adrenal cortex. So starting from superficial and going deep, you have the capsule on the very outside, then there's the zona glomerulosa, followed by the zona fasciculata, followed by the zona reticularis, and after or deep to the zona reticularis is the adrenal medulla. And what is released by the adrenal medulla? I'm going to start trying to be proper with what I'm saying. Right? Epinephrine and norepinephrine, adrenaline. And then you don't have to know the different regions of the adrenal cortex in terms of what they release. They release mineralocorticoids, glucocorticoids, and DHEA, which is essentially an adrenal androgen. It's how women get estrogen in their after menopause. It all comes from the adrenal glands. Cortisol and aldosterone. So where's cortisol released from? Fasciculata, and then what's in the reticularis is DHEA, and then what's in the glomerulosa? Aldosterone. But you don't need to know that. I'm not going to distinguish between those in terms of you knowing whether it's mineralocorticoids, glucocorticoids, or whether it's adrenal androgens. You don't have to know that. But you do have to know the layers. And how could I present that to you on something like a practical or a lecture exam? I could show you an image like this, and then maybe I could, and I'd probably do it on a picture because it'd be very difficult to do on a slide, and I could bracket them, right? from superficial to deep, deep to superficial. I could call it AB, ABC, something like that, but you do need to know the histological characteristics. You also need to know the location of all of these glands on like a model. Just the flat out, where's the pituitary gland, where's the thymus, where's the thyroid gland, where are the parathyroid glands, where are the adrenal glands, where's the pancreas. Just flat out, no histological, knowledge involved here. So where are the adrenal glands?
It's right above the kidney.
On top of the kidneys.
Kidneys.
In fact, if I pulled out a kidney model by itself, independent from the body, and pinned the gland on top of each kidney, that's what I'd be going for.
You want
It's
to
the
know
adrenal glands.
norepinephrine and epinephrine, though. Right?
Epinephrine and
You want us
norepinephrine
to
is
know
the
those.
adrenal medulla,
Okay.
and that's totally histologically and functionally different than the adrenal cortex. One is glandular tissue, and the other is just modified neural tissue. And epinephrine and norepinephrine are really neurohormones. So, alrighty. All right. So that's a lot over that, right? In terms of blood cells, identify the specific cell type indicated by the pointer. It's a neutrophil. How do you know it's a neutrophil?
It's
Multilobar, four-lobe.
So multilobar nuclei, that tells you right off the bat that we're dealing with a neutrophil. And then what's the stain like, kind of? It's pretty neutral, even though sometimes you get variations on that if you do like a single dip into basic. There's dips that you can do an actual staining procedure. It's called a quick stain, where it's going to pick up a little bit of, acidic stain, which stains red. It's pretty much neutral, and if I give you an eosinophil, it's going to be damn red, and you're going to see the granules clearly in it. So, everyone good with that? What are neutrophils' specialty? Bacterial infections. Can they target different types of infections than bacterial infections? To a lesser degree, yes, but they are the body's primary defense against bacterial infections. They have these little spear proteins that essentially poke holes in bacterial cell walls and lyse the cell. And they can also function as little suicide bombers, releasing a bunch of chest of enzymes to destroy bacteria. So, identify the structure indicated by A. That's the bicuspid valve. Identify the specific structure indicated by B. That's the papillary muscle. And then these guys here are called the chordae tendineae. What's the purpose of the valve, the chordae tendineae, and the papillary muscle? They all serve a specific function.
Unidirectional flow.
They prevent
backflow.
backflow. Right? They're all there to prevent backflow. The heartstrings in the papillary muscle, so when the ventricle starts to contract, what happens to the valve? It shuts because you're building up the pressure in the ventricle, so the valve snaps shut. In fact, what are your heart sounds? It's the closing of the valves. The first heart sound is the closing of the atrioventricular valves. The second heart sound is the closing of the semilunar valves. Lubbed up, because there's a sound associated with that, right? How many people got that? How many people would be able to tell me the function of a valve? What these guys do is they prevent backflow. They actually tense up to prevent, because otherwise it would just swing like a saloon door and you'd get the backflow of blood. It wouldn't be a very efficient system. You want unidirectional flow in the heart because you have specific areas of deoxygenated and oxygenated blood, and you don't want those mixing. Everyone good on that one? Does everyone kind of understand what I can ask about the heart? Have people, go ahead, Alex.
So they both prevent backflow?
All valves prevent the function of a valve in general, whether it be a valve in the heart, right, a valve in a venous system, a valve in the lymphatic system. All valves are there to prevent backflow. You want these systems to be unidirectional, right, because if they're going in the opposite direction, you're mixing either lymphatic fluid or blood that's oxygenated or deoxygenated, and it decreases the efficiency of the system. Everyone good? Identify the vessel indicated by A. Say that louder, Whitley Safrone.
The left common carotid artery.
Did everybody hear her? Does everybody agree with her?
Sure.
Say it again. Left common.
So, what is this vessel right here specifically? Be specific with me. It's the ascending aorta. And then this area here is the aortic arch. And then you have the first branch off the aortic arch is the brachiocephalic trunk. The second branch off the aortic arch is The left common carotid artery. The third branch off the aortic arch is the left subclavian artery. So, when I have this guy on a practical, doop, 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 right? A, B, C, is that an easy thing for me to do?
Yeah.
Yes, it is. It's a very easy thing for me to do. Do I want you to know the branches coming off of the aortic arch?
Yeah.
Definitely do, because these branches feed the entire top 1/3 of the body. Everyone good? Okay. So that answers B. What's B?
Aortic arch.
Aortic arch. Alrighty, identify the cell type indicated by A. A lymphocyte. How do you know it's a lymphocyte and not a basophil? It's got a halo. The halo is the telltale sign of a lymphocyte. And a lymphocyte is considered a granulocyte or an agranulocyte? An agranulocyte, meaning that there are actually granules. They're just not big enough to be seen with a light microscope. So you can't really see the graininess in the cytoplasm, which are actually digestive enzymes. They're proteolytic and antibacterial enzymes that are in the cytoplasm of white blood cells. And then you can start distinguishing between B and T lymphocytes, but we're not going to do that. Identify the cell type indicated by the pointer.
Erythrocyte.
An erythrocyte, aka
Red blood cell.
Go ahead, Daniel.
This might be a little bit picky, but on the previous slide with the blood cells, we're saying identify like the specific cell type, and this one was just like identify the cell type. So you could think to say like leukocyte as opposed to like whatever that was the.
How many people said leukocyte on that?
What if I put both too because I was confused? One was
It looked
specific
like you were
and
just
one was just
identifying
cell
the difference
type.
between a white blood cell and a red blood cell.
On a practical, if you get an identify the cell type question, what are you going to answer? Try and be as specific as possible. This one, I will give it to you. If the terminology confused you, I will give you that one. Right? If you said, how many people said leukocyte again? How many people didn't know what cell type this was? Well, I'm never going to know that because now So on this one and on this one alone, I will do that. Alright. Identify the vessel indicated by the pointer. Brachial artery. Into which two vessels does this vessel feed? The radial and the ulnar. And what's feeding? How do I know it's feeding? In which direction are arteries carrying blood? Away from the heart. So when I say feed, right, it's the movement of blood is always going to be going away from the heart. If it was drain, it would be a venous system and it would be going back toward the heart or back to the heart. How many people got that one? Brachial artery and then radial and ulnar arteries. Where do you take somebody's pulse?
Your radial.
The radial artery. The radial pulse.
Unless you can't find it there, and then where do you take somebody's?
You can take it at either carotid or femoral, depending. Alrighty.
How well you know it.
That's how well you know them. I prefer femoral. I'm taking the pretty lady's pulse. Alrighty. I'm kidding. I'd go with carotid. Identify the chamber indicated by the pointer in the image. First off, how'd I ask the question?
Chamber.
Chamber. So what do you know I'm looking for?
A chamber.
I'm looking for a chamber of the heart. I'm looking for one of the atria or one of the ventricles, right? And which one is this? It's the left ventricle. How do you know for a fact that's the left ventricle?
Thick
The
myocardium.
thickness of the myocardial wall, because the left ventricle pumps to systemic
Systemic.
circulation and the right ventricle pumps to
Pulmonary.
circulation. So the left ventricle has to pump fluid through a lot more pipes and a much greater distance of piping. How many people got that one? Everybody? Diane did too. She's doing
Good
well in this
review.
class. She's doing very
I
well.
normally don't think below the collarbone anymore.
Okay, first off, is this an artery or a venous system that we're looking at, and why? It's a venous system. Do I see an aortic arch here?
No.
No. In fact, where are these two vessels going back to?
The heart.
So this is, yeah, but specifically, so this is the superior vena cava, that's the inferior vena cava. Where are they draining blood back to specifically? To the right atrium. So you know that this has to be a venous system that we're looking at. So, identify the vessel indicated by pointer A. So the vessel indicated by pointer A is the superior vena cava. The vessel indicated by pointer B is the
Jugular? Jugular vein?
jugular.
Jugular vein?
The jugular. Everyone good? And if you're not, then you know what to study. Now, let's come on over here for a second. Where could I ask that on one of these models? And in fact, the one that David moved, Rod, where would the jugular vein be on that? So there we have the jugular, and then you have the internal and external. Where's the branching of the internal and external jugular on that model? That's a very good model to show it. Right there. Right to the side. So definitely think about the models that I can show different vessels on, because that one's ideal for a lot of things. And then you have the jugular. And where does the jugular drain into? What's that point right before the superior vena cava that the jugular drains? So we'll go to the subclavian, and then the subclavian is going to be, and then from the subclavian it'll drain into the brachiocephalic, and from the brachiocephalic it'll drain into the superior vena cava. Does that kind of make sense? Yeah?
Where's the brachiocephalic on that?
On this guy it would be right here, right at this point. Now the subclavian, there's actually another vein that drains, and technically the 2 points, it's the point after the other vein that drains in that demarks the subclavian from the brachiocephalic. But on this I'm just going to call, actually I'm going to eliminate ambiguity and say that the jugular drains directly into the brachiocephalic. I'm going to ignore that subclavian distinction. We're going to call that the distinguishing point, because the vein that you need to know is not there. Or the vein that actually makes that demarcation point is not on any of these models. So, jugular drains directly into the brachiocephalic, which drains into the superior vena cava. Everyone good? Does that make sense how I can approach asking this? So what else could I ask on this particular image? I like this image. I like that it's not color-coded. I like a few things about this image. What else could I ask? What about the C? Had I asked C? So there's 3. It would be the cephalic, and isn't that weird that it's called the cephalic? It's probably because it drains into the brachiocephalic, but you would think that something called the cephalic vein would be draining blood from the head. Alrighty, and then this one running deep through the middle is the brachial vein, and this guy in here is the basilic vein. And then this guy right here, where it runs across the anterior portion of the elbow, is called the median antecubital, and I want everybody to know that one. How come? Because that's where you draw blood from. And then the median antecubital is drained into by, if there's a radial and ulnar artery, there has to be a radial and ulnar vein. You know what I mean? So how are you going to approach studying this? Well, let's look at some of these models. So what can I point out? The same exact things that we're talking about up here on these models. You just have to know the vessels. I'm not going to sugarcoat it. There's 26 arteries. There's 27 veins that you need to know. And I can pin any of them on the constellation of models we have here. For example, what vessel would that be? Identify the vessel indicated by the pointer if it's running through this area right here. That would be the brachial artery. It's just a different way of looking at it, but it's the artery that's running through the arm, right? It's the artery that's running through the brachium. So there's all of these models up here I can actually pull out and ask about. You know what I mean? That model that David moved over there was also on this table a minute ago. These are potentials that I can ask on a practical, and are they going to be peppered around the room? They have to be. It's the only thing I have at my disposal, right, to test your knowledge of the material. So yeah, I'm going to be using all of these for like the carotid arteries. Would you hold this guy up for a second? What would be a logical choice for me to ask about the carotids?
The internal and external.
So you have the common, and then the one that's running externally would be the external carotid, and the one that's running internally would be the internal carotid. And what's that one that's actually running through the transverse foramen of the cervical vertebrae there? What's that one called? That's the vertebral artery. Is there any way to get out of this without knowing it?
No.
None. Zero. You're going to have to painstakingly sit down and memorize these things. There's just no other way around it. Everyone good?
Well, we have the vertebral artery again.
Go ahead.
The little guy.
It's the one that runs through the transverse foramen of the cervical vertebrae.
Like turn around. Yeah. Running through the running.
I would just call it that. But you're right, it's just such a weird way to identify the cell type indicated by A. So it's an eosinophil. I'm not going to take leukocyte again. Maybe I threw in specific because I was doing this quickly, but we went over these. We did a differential white blood cell count in class, right? I'm definitely looking for the cell types, the different types of white blood cells. Go ahead.
This right here.
Huh?
External. That's the carotid artery.
That you're feeling, that would
Is
be
that
the common.
That's the common?
Yeah.
Okay.
Okay, everyone with me?
What was the answer to A?
Eosinophil?
Oh, here? Oh, yeah,
The
eosinophil.
E one, right? Okay.
So it's an eosinophil, and what are eosinophils very good at targeting? Parasitic infections, things that have the same cellular machinery as us, in other words. When you're talking about like a tapeworm and you break it down to the cellular level, the machinery of a tapeworm on a cellular level, meaning that they have membrane-bound organelles and a nucleus and they carry out, cellular respiration, is very similar to us. So it takes a unique set of unique complement of, for example, proteins to kind of target and destroy those. What else do eosinophils do?
Allergic reaction?
And it's anti-allergic. So what are they now doing to treat things like autoimmune diseases? They're introducing parasites. They're introducing things like tapeworms because it cranks up the number of eosinophils, and they produce a natural antihistamine and anti-inflammatory that runs systemically, and it's had a lot of success in clinical trials. In fact, that's one of the reasons that they think that there's such an abundance of autoimmune diseases in the Western world, is because we don't have eosinophils anymore, because we've gotten rid of all these parasites that we've kind of evolved with. So now we've opened up a new constellation of problems.
So the guy who gave himself ringworms to help with allergies, that's the same thing?
Exactly. Because a ringworm is just a parasitic infection, and somebody who has ringworm, if you were to run a differential white blood cell count on them, what would you expect to find elevated? Eosinophils. So eosinophils are anti, right? They produce antihistamines and anti-inflammatories. What's the one that produces inflammatory compounds like histamines? Basophils. So when would you expect to see basophils, right, elevated? During an allergic reaction. Your professor is so tired of this semester that he decides to run naked through a field of poison ivy, right? What would you expect to find elevated on a differential white blood cell count? Basophils. Alrighty, so does that make sense? Specific structure, and how do you know? Indicated by?
B.
B. So there are two parts to the hypophysis, right? What are they? Anterior and posterior. And how can you always determine the anterior pituitary gland or the adenohypophysis from the posterior, the neurohypophysis? It's
Size?
bigger. It's relative size. So B is indicating the
Anterior.
pituitary gland.
Adenohypophysis.
Yep? Alrighty. Identify the structure indicated by A. Tricuspid valve.
Tricuspid.
Try before you buy. So you could also call it the right atrioventricular valve. Right atrioventricular or tricuspid is fine with me. And identify the vessel indicated by B.
Pulmonary artery.
Pulmonary artery. What's the oxygen status of blood in that artery?
Deoxygenated.
Deoxygenated. So it's the only artery in the body that carries deoxygenated blood. So it kind of throws this idea that arteries always carry oxygenated and veins always carry deoxygenated blood on its head. Why do you think I took the color coding away? So on a practical, if I asked you, right, you recognize where it's going. So these are going, it goes pulmonary semilunar valve to the pulmonary trunk. Trunks always branch, right, just like the trunk of a tree, and then this branch is the pulmonary artery. So then I could ask you a question, for example, into which circuit is this pumping?
Pulmonary circuit.
The pulmonary circuit. The oxygen status of the blood? Deoxygenated, right? Because it's going to the lungs. And what's it going to pick up in the lungs?
Oxygen.
And what's it going to ditch in the lungs?
Carbon dioxide.
Carbon dioxide. And then these guys here are the pulmonary veins. What's the oxygen status of blood in this particular vessel? Oxygenated. So now veins are carrying oxygenated blood, right? That's typically not the pattern in the rest of the cardiovascular system. Arteries typically carry oxygenated blood and veins typically carry deoxygenated blood. But the bottom line is an artery takes blood away from the heart, a vein brings blood back to the heart. It doesn't have anything to do with the oxygenation status of the vessel, right? Everyone good? Does everyone kind of see questions that I could ask about that? Alrighty, so in terms of studying for the lymphatic system and the cardiovascular system and things like that for an exam, let's look at the lymphatic system models that I have. So lymph nodes and things like that we're going to look at in class today, and we're going to go over PowerPoints, and I'll probably just give you kind of images like that in terms of what I give you on a PowerPoint. I'm not going to do very many of the histological images in terms of the lymphatic system. This guy, this model here is called lymphatic man. Right? When you're looking at these lymphatic structures such as lymphatic vessels and the lymph nodes and the different beds or lymph node beds like the inguinal lymph nodes and the intestinal lymph nodes and the cervical lymph nodes and the axillary lymph nodes, know that you're going to have to distinguish those because is it going to be indicative of a different type of infection if certain lymph node beds become inflamed, right? Or they become enlarged? Yeah, it is. Like if you had strep throat, which ones would probably pop up? The cervical lymph nodes, as opposed to if you have gonorrhea, which ones are probably going to pop up? The inguinal lymph nodes. And if you have a systemic infection, right, like the flu, you get all of them because it's kind of floating through everywhere. So you definitely have to be able to identify the different lymph node beds. You have the left lymphatic duct here. You have the spleen, you have Peyer's patches and lymphatic nodules. So this guy is the lymphatic model. It's the best lymphatic model that I have, and I may even have 2 stations on it because I just don't have very many lymphatic models. I can show you what's this guy right here?
Spleen.
The spleen, right? I can show that on a few models. And then histological images we're going to go over in lecture today. You know what I mean? So just kind of go down, who printed out their list? This is the list from the lymphatic system. Study models and charts to locate the following groups of lymph structures. Lymphatic ducts, right and thoracic. So there's the right and left lymphatic duct. The left lymphatic duct is interchangeable with the thoracic. Everybody has a thoracic or a left lymphatic duct. Only 15% of people have a right lymphatic duct. So it's presented in textbooks like that's just, it's standard that there's right and left. It's not. Most people only have a left or a thoracic lymphatic duct. You can call it the left lymphatic duct, right, if you want, or you can call it the thoracic lymphatic duct. It doesn't matter to me. There we go. Lymph nodes, cervical, axillary, inguinal, iliac, and abdominal. What are you going to do? What's the model to look at? Lymphatic man. Individual lymph nodes, cisterna chyli, that's the enlargement at the beginning of the left lymphatic duct, or the thoracic duct. They're just 2 different words for the same thing. Lymphatic collecting vessels, what are all these guys out here? They're lymphatic collecting vessels. So this is really my primary guy. In terms of the tonsils, Identify secondary organs of the lymphatic system on models or diagrams. So what's this guy? Spleen. Where's the thymus? Does anybody, so would you grab that heart model real quick? So the thymus, go ahead and point out the thymus, Carmen. It's right on top of the heart, whereas the thyroid is up just inferior to the larynx, right? So, pharyngeal tonsil, palatine tonsils, lingual tonsils, and tubal tonsils. Where can I point out the tonsils at? Well, this is one model I can kind of point them out at. Has anybody seen that tongue model?
Yes.
Where is that tongue model? Yeah, would you, it's right there. Go ahead. Yeah, perfect. So where would we have the valate papillae, and then where would the lingual tonsils be? When they look in your throat for your tonsils and they say, wow, your tonsils are really inflamed, they're on either side of what's called the pharyngeal arch, so either side of the opening to the throat, essentially. Those are the palatine tonsils, right? So the pharyngeal tonsils, aka the nasopharyngeal tonsils, are going to be located further up, so let's see if I have a model. But do you see kind of the thought process that I'm going through with looking at these, right? Is there a model for that?
No.
Maybe pull that one over there. See that big nose model?
The sinus one?
Yeah, that guy right there. The nasal cavity model. Oh, here's one. So there are your pharyngeal tonsils right there. Because the palatine tonsils are located to either side of the throat. They're the lingual tonsils right there. So these are indicating lymphatic tissue. Here are the palatine. So in other words, I would ask tonsils on this model right here. I don't know if it really shows them on that model. Will you pull off the cartilaginous and bony septum there? No, not great. Not great. In fact, the only model that I really have to ask those on now that I look at all of these are this guy. So I go through this exact thought process when I look at all of these things. You know what I mean? I just go, which models in this classroom can I use to ask about that? Model for the tonsils is going to be this one right here because it shows 3 of them. Right? That tongue model, lingual tonsils, it's also for the special senses. What else can I show on it? So that's kind of the thought process to go through with all of this. We just ran through the lymphatic system, pretty much. Without the histological images, which we're going to be looking at in lecture today, we just ran through everything you need to know on the lymphatic system. Everyone good?
How will
All right.
you use this one? Because this one seems confusing.
Yeah, what could I use on this one? So go ahead and hand it to me. What is a portal system? Somebody define portal system for me.
Capillary to capillary.
Yeah, it's when one capillary bed is drained into another capillary bed, and there are 2 primary portal systems that we're concerned with. The hypothalamic hypophyseal portal system, because that takes blood directly from the hypothalamus to the anterior pituitary gland, which carries tropic hormones from the hypothalamus to the anterior pituitary that says, hey, release this hormone in response to whatever stimuli. You need to know ACTH, right, and the thyroid stimulating hormone. And the other portal system, the hepatic portal system. Why is all the blood from the GI tract drained directly to the liver before it enters systemic circulation?
Filtration.
To be filtered. It's a filtering mechanism. You don't want plant toxins and things like that just running willy-nilly into your cardiovascular system and perfusing all your tissues. So when you look at this liver here, this liver is actually true to size. This is not an enlarged liver. It's not like made bigger, right? Your liver is about that big and it sits like this. So when you look at this guy, right, you're looking at the model in a weird orientation. So the liver sits like this up under the rib cage, and it's about this size, right? This is not an exaggerated model. So this guy here would be the hepatic portal vein. The hepatic portal vein branches out, and then what type of capillary is in the liver? The sinusoidal capillary. So blood actually escapes and they percolate through these little canals in the liver, and they're actually filtered out, and then you have the hepatic veins that collect that blood. The hepatic veins will drain directly into the, what's this big guy right here? So these are the common hepatic veins, or the hepatic veins, and then the hepatic veins drain directly into the
Superior vena cava?
the inferior vena cava. So this
Oh.
is the inferior vena cava right here.
Yeah.
Why does that model have green, purple, blue, and red vessels?
Yeah, why? What indicates? So the green vessels are probably going to be associated with bile, and by probably I mean that's the convention to point out bile. So what you're looking at there is bile ducts, and when we actually study the structure of the liver in terms of the hepatic cells themselves and the way that they're arranged, right? That will make more sense. But you're looking at the flow of bile there. So the purple, what does the purple indicate? The hepatic portal vein is carrying blood directly from. So all the blood from the GI tract is drained, and the common drainage point is the hepatic portal vein. The reason that it's purple is to indicate that it's nutrient-rich blood. That's just a convention for nutrient-rich.
Then
And
why
be-
wouldn't it be red?
Because that's
Okay.
oxygen-rich, right? So does that make sense, the different conventions? And then when we get into studying the common bile duct and the, cystic duct and the left and right hepatic ducts for bile, right, that's when we're going to start studying the green stuff on this. Because what is this guy right here?
Gallbladder.
It's the gallbladder. Does this model make sense to everybody now?
So the important ones are the hepatic portal vein, inferior vena cava, and what was the third one?
So the hepatic portal vein will drain blood from the GI tract, right? So it goes from one capillary network into another capillary network in the liver, and that's what makes it a portal system. It's then going to percolate through the liver, and it's going to be collected up by the hepatic veins. The hepatic veins are going to converge on the left and right hepatic vein, which are going to drain directly into the inferior vena cava, which is going to be taken back to where? The right atrium. Does that kind of make sense? So yes, there's a lot that I can ask on this model. How many of these models do I have? I have one. So how much can I actually ask? There's a lot that I could potentially ask, but how many stations are going to be dedicated to this model right here? One, unless I can make a clone of this model, right? I don't think we have another one of these. Just like this guy, when I ask you about hepatic portal circulation in the chair, this is probably the best one to ask about the superior mesenteric, inferior mesenteric, right? And splenic veins, because it's so clearly shown on this. So when you're going through and you're like, gosh, how can he ask about that? Raise your hand and ask me, because there's models that are ideal for showing certain things. But definitely no, go through and just learn them all. There's no way to sugarcoat this. There's only so much window dressing you can throw on anatomy. You just have to rote memorize these things. Yeah? Go ahead. So kidney. So identify the vessel indicated by the pointer. Identify the vessel indicated by the pointer.
Renal vein.
Because it's feeding the kidneys.
Kidneys.
So things like that. Does anybody have any questions about the approach they should take to studying this? No? Everybody good?
Mm-hmm. Mm-hmm.
Okay.